today our topic is vulnerability. So why some get addicted and majority not? So this is the brief outline of my presentation. First, we'll discuss about the genetics. So why we need to discuss about genetics? So addictive disorders are etiologically very complex conditions. Without any doubt, millions of people are exposed to addictive agents each year, but vast of the majority are not addicted, though they may develop tolerance or even a brief period of dependence to some of the agents. And this example is much more true now, because in many of the cases of cancer chemotherapy, we are using opioids, and many of them may become tolerant to the dose, may develop withdrawal symptoms, but very few of them actually in long run developed so-called dependence or addicted to the substance. Why is it so? So from to searching for this answer, so there are many models which are very much interrelated. One of the important model is your stress diathesis model, so which was first given by Paul Mill. So according to him, some of the individuals are predisposed or diathesis. They have some diathesis, that is vulnerability. Now, with those vulnerability, if they are exposed to some certain amount of stress, they will break down. So this is true for many of the other psychiatric illness, and it's also true for addiction. So another important related factor is gene and environment. So here gene represent that diathesis, and another related model is biopsychosocial model that has been given by George Engel. So that is also a very important model. Here George Engel told that better not to see those stressor as stressor, include it as one of the vulnerability, like psychological vulnerability or social vulnerability. For to causation of any kind of illness, there may be some biological vulnerability, some psychological vulnerability, and some social vulnerability. Reach the actual etiology of any kind of disorder. So with this introduction, I will briefly discuss about genetics of alcohol, of addictive substances. In genetics discussion, we should proceed from family study. Then next, we should go to twin study, then adoption study, then newer studies like linkage analysis, then we'll go to candidate genes. And most recently, which are used is GWAS, genome-wide association studies. So I'll describe very briefly about all the studies that have been done in addiction. Like family studies, it has been found that male children of an alcohol-dependent parent have 4 times the risk of becoming a problem drinker compared with the children of non-dependent parents. While female children of alcohol-dependent mothers evidence a 3-fold greater risk. If we go to twin study, we'll find that among twin pairs in which one twin was diagnosed with alcohol dependence, there is a significant difference in the proband concordance rate among monozygotic and dizygotic. So both are increased. In monozygotic, it's around 54%, and in dizygotic, it's around 31%. Now, this monozygotic and dizygotic ratio is very much important to give a model, because clearly addictive disorders, any of them, be it alcohol, be it opioid, be it cocaine stimulant, they are not due to a single gene, that is for sure. So what is the actual hereditability? So there can be 2 models. One is polygenetic and another can be heterogeneity. So in polygenetic model, what we used to see is that polygenetic model, there are many genes that needs to be there co-occur, and they leads to a certain illness. But in heterogeneity, there are few genes of moderate to high effect size. So even 1 or 2 genes, if they co-occur, can lead to one disease. In those cases, like in autism, we will see monozygotic is to dizygotic ratio is very high. So it's polygenetic. So In those cases, there are many genes. If they co-occur, they can lead to one disease. But if there is even a slight variation of genetics, that will not lead to autism. But it's not the case in addictive disorder, because the ratio is around, as we have seen, around 2:1. So there are some high effect size gene that even the 2 or 3 genes can lead to one certain disorder. So if you see the heritability, it ranges from it varies from study to study. It's around 40 to 90%. The more according to this study meta-analysis, the highest heritability was found with cocaine, around 70%, and lowest with hallucinogen. And surprisingly, if you can see that gambling has higher heritability than even cannabis and sedatives, which are well-established addictive disorder. So this is one of the cause why gambling has been included as one of the addictive disorder, because of a very tangible heritable factor. It's not only the environmental thing. Another important finding is that more chronic and severe form of substance dependence showing greater estimates of heritability. Next, we'll move on to adoption study. So subjects who had biological parents with severe alcohol problem were significantly more likely to have alcoholism themselves than if their surrogate parents were alcoholic. And Kluninger, with their famous study on your Swedish adoption study in the 1970s and 80s, they have showed that significantly higher rates of alcohol abuse in adopted sons and biological fathers registered with alcohol problems. So all these things actually directing towards some genetical basis of alcoholism. Next, we'll move on to linkage study. One of the largest linkage study that is there is COGA, that is collaborative of genetics in alcoholism. So that collaboration of the study, they have found that in long arm of chromosome 4, that is linked to alcohol dependence, and they have compared this long arm 4 regarding some of the phenotypes that are consistent with alcohol dependence, which is delayed P300 and maximum drinks in 24 hours. All those things are very much linked with that region. Later on, they got to know that that region is actually synthesizing the protein ADH, alcohol dehydrogenase. So before we discuss about candidate genes, we need to discuss about the metabolism of alcohol, because all the candidate genes which has a very significant evidence are linked to this alcohol metabolism. So alcohol from used to get metabolized to acetaldehyde, after that acetate. So alcohol to acetaldehyde is by alcohol dehydrogenase, and acetaldehyde to acetate by aldehyde dehydrogenase. If this acetyl dehydrogenase somehow got accumulated, it will lead to unpleasant reactions, which is called flushing reactions, alcohol-induced flushing reactions. So along with that flush, you can have headache, palpitation, hypotension, itching, rash, all those things. So there are variants of ADH and LDH. So first we'll discuss about ADH. At least 7 different genes synthesize ADH. Among that, most important is class 1. Class 1 genes have 3 variants, A, B, and C. Among that, these 2 alleles that I have mentioned, ADH AB2, 1B2, and ADH 1C3. These 2 alleles are noted to be protective develop alcohol dependence. So you can say in reversely, you can say the other alleles are actually more vulnerable than these genotype persons of becoming alcohol dependence. Why is it so? Because these alleles are having high metabolic rate of alcohol, so they tend to accumulate acetaldehyde. That's why there is a high risk of developing that flush reaction. Next, the most important is this mitochondrial aldehyde dehydrogenase 2. So there are 2 variants of allele. One is LDH22 and LDH 2-1. So if LDH, it can happen in three possibilities there. One is LDH 2-1 homozygous, LDH 2-1 and LDH 2-2 heterozygous, and then LDH 2-2 homozygous. So in case of the first variant, 100% enzymatic activity is there. In case of the second variant, only 17 to 38% of enzymatic activity is there. In case of third variant, only 0% enzyme activity, almost none enzyme activity is there. If the LDH is having no action, so all the acetaldehyde will, and there is a high risk of flushing reaction. So this genotype is very much prevalent in Eastern Asian populations. So that's why they are much more protected against alcohol dependence. So we can say contrary that this genotype is much lesser in case of European and Americans. So they are, compared to this Eastern Asian population, they are at high risk. Next, we will move on to JIVA study. So as we all know that in addiction, JIVA studies is in its infancy. Only few studies has been done. But as a study, you need not have any hypothesis before starting any JIVA study. And you can compare n numbers of single new SNPs. Only 3 or 4 gain has been there in JIVA studies in addiction. One of the important one is Tobacco Genetics Consortium study, and also by Liu et al. They have found that nicotine acetylcholine receptor subunits are alpha 5, alpha 3, and beta 4. It has a cluster. It used to happen in a cluster. So this cluster polymorphism, especially missense polymorphism, is very much significantly associated with tobacco addiction. And not only that, these 3 different locus are additive. Like if one person is having only one, the risk is lesser than if the other person is having 2, risk is lesser than the person who is having all the 3 polymorphism. Other JIVA studies that has some significant effect is that cytochrome 2A6, which is related to metabolism of nicotine to cotinine. Another is dopamine dehydroxylase. Only one significant result has been found in alcohol in JIVA studies, which is autism susceptibility candidate 2. Now we'll move on to the relationship between nature and nurture. So as we have already discussed, there is genetics and environment, because even if you are very much predisposed to alcohol, if in your environment that substance is not there or some stressor is not there, you will not become addicted to alcohol. So what are the other factors? There are some very good study regarding that, but before that we have to understand the dopamine system and alcohol. Decreased striatal dopaminergic function associated with altered sensitivity to natural reinforcers. So this increases the vulnerability to use reinforcing substance such as alcohol and other addictive substances. So within ventral striatum, like nucleus accumbens, dopamine pathway, often referred to as the reward pathway, as you all know, reduced dopamine receptor densities have been observed in alcohol-dependent patients. You all know about COMT. COMT is one of the enzymes that metabolize dopamine. So it's actually responsible for degrading dopamine. So there is in 158 position of this enzyme, there can be valine or valine methionine or methionine. There are 3 polymorphisms. So this methionine polymorphism has 3 to 4-fold reduced activity of COMT enzyme due to reduced protein stability. So what is happening? If COMT activity is reduced, metabolism of dopamine is reduced, so more exposed to pleasurable effect of dopamine. So met associated with increased drinking. This is one of the established thing in case of alcohol, but it's not true for some of the other addictions. Now, relationship between childhood adversity score and alcohol dependence. This is one of the very important study. Now, if you see among these 3 polymorphisms, starting result, if you see, if there is no childhood adversities, almost there is very less chance of the less probability of having alcohol dependence in all three cases, but more so for met allele. When that exposure is crossing the mid exposure, that is either moderate exposure or severe exposure, the chance of probability of having alcohol dependence is much more higher for met allele compared to others. So this is how the environmental factors and genetic factor is interacting with each other and causing the consequence of alcohol dependence. Apart from genetics, now briefly we'll discuss about other individual variabilities like personality. According to temperament character in inventory, if we say go, some of the personality variability are increasingly associated with substance use, like high novelty sensation seeking and low harm avoidance and reward dependence. Similarly, some of the temperament variables that predict later substance use, like high emotionality, low attention capacity, low sociability, impulsivity, all those things. Difficult temperament like that Thomas and Chase criteria, if we go, some are very much predisposed to develop substance use in future, like high activity level, low flexibility, low task orientation, mood instability, social withdrawal. All those adolescents are much more vulnerable to develop addiction in future. One of the research study also found difficult temperament is much more important than the family history of alcohol itself. According to big five factors, as we know that we used to remember by Ocean. In this case, the positive alcohol addiction is positively associated with openness to experience and negatively associated with agreeableness and conscientiousness. This part, I think Vivek sir has described in detail. I'll just touch upon this. Apart from all the vulnerability, adolescent itself is very much vulnerable. As you can see in this graph, the first exposure of drug is mostly during the period from 16 to 20. After that, it will decrease, and before that, also it's not that significant. And if they are exposed to this initial period, like from the first graph, the A graph, you can see if a person is exposed before the age of 12, he is much more susceptible to develop alcohol dependence in long run than if the person is exposed after 19 years. So not only this age is vulnerable for initiation of substance use, but after initiation, the development of dependence is also very much common in this age group. So among other individual risk factors, perceived sense of safety and perceived sense of high outcome from the substance, external locus of control, external disorder, internal disorder, poor self-regulation, which will actually the extent of external disorder only, poor social skill, which is actually we can say the consequence of externalizing or internal disorder in adolescent. All these things are actually can be regarded as vulnerable factor for future dependence. So in internalizing disorder, the PPT is there. I will not read out all those things. There's very few studies that has been done which showed prospectively that with childhood history of internalizing disorder, the substance use will be more. But there are very significant amount of association between comorbid association between internalizing disorder and substance use. That also we have seen in today's case, especially in case of PTSD and in case of sexual victimization. So this kind of adolescent with abuse, they are much more, almost 5 to 6 times more vulnerable than the others developed substance use in future. So this is another chart that's showing that a significant amount of high prevalence of mood disorder, anxiety disorder, and personality disorder with any kind of other substance use. So external disorder, no, not go into detail, but I will just quote some of the studies. Like one 10-year follow-up study of monitoring children into young adulthood showed that PhD subjects were almost 1.5 times more likely to develop substance use disorder compared to control. And another comparative meta-analysis of 13 studies showed that ADHD was associated with alcohol and drug use disorder in adulthood and nicotine use in adolescent. And this study, the last study that I have mentioned, has been done in Australia. They have found that the conduct problem during the first 8 years of life, and when this population was prospectively examined, they have found that almost more than 2 times increased chance of alcohol-related problem, more than 3 times chance of cigarette smoking, and almost 3 times of illicit drug use. So apart from these individual factors, there are some very important family factors which can be considered as stress, but I would like to consider it as risk factor only, because some of these things are very much modifiable, like loss of parents. It's not modifiable but can be replaced. Interpersonal distress between family members, and very poor communication between parent and the child, and parent or sibling having substance use, not only having genetic predisposition but also the very important learning theory is this modeling is also a very important part of it. Absence of clear rules and consequence in the family, and authoritarian parenting. It is more demand and very much less responsiveness. This actually is very much associated with future substance use and other external eating disorders. Now, in community risk factor, lack of academic expectations, indifferent attitude of teachers, and bullying and violence regarding the school context. These are very much important regarding the school context. So these factors are regarded as some of the important risk factor in school context. Apart from that, peers, who are the peers actually determine the adolescent substance use. So if the peers are engaged in antisocial behavior or other substance use, there is much more likely chance that the patient or individual will also become a substance user. Apart from that, youth in labor force, like childhood labor, and availability of alcohol or easy availability of alcohol or drugs in a neighborhood, and neighborhood disorganization like poor sanitation, poor livelihood, all those things are actually can be considered as community risk factor. The next question is, why you are making so much fuss about this vulnerability? What will you gain from learning this vulnerability? Many things to gain from it. First and foremost, I would point out is the prevention. And I don't think prevention has been a part of this foundation. So I'll just briefly take another 5 minutes of time to describe about the prevention in substance use. Apart from the prevention, another thing that this vulnerability, learning about vulnerability can do is that we'll get to know why this person is like this. What are the antecedent of that? Why he became like this? Why he is not? We are not able to change him. He is behaving like that. We'll find some etiology. We'll find some empathy that person. So we'll have much more empathetic approach towards the patient if we learn the vulnerability. Not only that, in clinical aspect, to take care of perpetuating and maintaining factors, as we have seen in today's case conference. One of the perpetuating factors is externalizing behavior, another of the perpetuating factors was internalizing behavior. So if we get to know about the vulnerability also, because many of these vulnerability also continued as perpetuating and maintaining factors. So if we can treat those factors, we can have a good outcome. So next we'll discuss about prevention. So as you can see in this box, that table, that for any kind of prevention program, our approach should be to make that probability of substance use from high probability to low probability. For that, what we have to do is that we have to decrease the vulnerability or risk factor. At the same time, we have to increase the protective factors or resilience. Okay. So according to Institute of Medicine, there are actually 3 types of prevention that can happen. One is universal, selected, and indicated. Now, universal prevention is actually address the entire population without having any filter. It's applicable for all of the population in the society. Selected intervention are only for those target subset after that some risk assessment should be done. After that, at least only that subgroup will be intervened. Indicated population is that either those population are having early signs of that disorder, or they have some diagnosed vulnerability of those disorder. Like universal population prevention, how we will use our knowledge of vulnerability in universal population, universal prevention. We can use, like we have already discussed, that adolescence itself is a very much big vulnerable factor for substance use. So as we can see that many of our bill, like bill or act, like COTPA Act, all those things, they are trying to do is that to delay the age of onset of substance use. They are stopping or banning the sale of substances in and around school campus. They are stopping or banning the sale in adolescent below the age of 18. So by all these things, they are actually delaying the onset of substance use. So that is one. Next is selective prevention. So in this prevention, at-risk population or vulnerable population are selected, then we will intervene into that. So this changing access with the environment of those population, like you can take at-risk population as some of the population who has dropped from school, like one of the case that has been discussed. If say this kind of children have been intervened at the right time, then we may be able to stop this substance use in the future. Like changing the access within the environment, promoting the development of personal and social skills so that they can be much more successful in occupational region of the life, promoting positive relationship with families, addressing social influences and providing social support. Next and most important from our perspective is indicative prevention, like management of externalizing behavior to prevent addiction in future. I will mention one very important study. This meta-analysis showed that 674 medicated individuals, that is medicated with methylphenidate, who are diagnosed with ADHD, and it was compared with 360 unmedicated subjects, and they have followed them for 4 years, and they have found that there are at least twofold reduction of risk of developing substance use in medicated population than unmedicated. So this is really a very positive aspect, I would say. So I think Dr. Arun already discussed this approach. So this is the approach we used to for assessment purpose, you should always progress from vulnerability to psychopathology to consequence. But in case of management, you should first address the consequence. What are the consequences of substance use? Then the substance use behavior itself, then vulnerability. Thank you.